Hello friends and welcome. Today we shall talk about electrophoresis. In today's deliberation, we shall discuss different aspects of electrophoresis and its various types. Let's first understand what electrophoresis actually is. Electrophoresis is the migration of charged particles or molecules in a medium under the influence of an applied electric field. Any electrically charged particle dissolved in aqueous solution when placed in a constant electric field will start to migrate towards the electrode bearing the opposite charge. The speed of the particle movement will be directly proportional to the applied voltage and particle charge, but inversely proportional to the particle size. The common purpose for carrying out electrophoretic experiments include determination of the number, amount and mobility of components in a given sample, and or to separate them, and to obtain information about the electrical double layers surrounding the particles. Let's firstly highlight the principle of electrophoresis. An electrophoretic separation is performed by injecting a small band of the sample into an aqueous buffer solution contained in a narrow tube. A flat porous support medium such as paper or a semi-solid gel. The high voltage is applied across the length of the buffer by means of a pair of electrodes located at its each end. This field causes ions of the sample to migrate towards the opposite charged electrodes. The rate of migration of a given species depends on its charge and its size. Separations are then based on differences in charge to size ratios for the various analytes in a sample. The larger this ratio, the faster an ion migrates in the electric field. Now let's discuss the types of electrophoresis. Electrophoresis is broadly classified into two types. That is, number one, free electrophoresis or electrophoresis without stabilizing media, and number two, zone electrophoresis or electrophoresis in stabilizing media. Firstly, we will discuss free electrophoresis. Free electrophoresis, also known as carrier-free electrophoresis, is a matrix-free electrophoretic separation technique. Free electrophoresis is an analogous technique to capillary electrophoresis with a comparable resolution that can be used for scientific questions where semi-preparative and preparative amounts of samples are needed. Free electrophoresis is further divided into two main techniques, that is, microelectrophoresis and moving boundary electrophoresis. Microelectrophoresis. It's an electrophoretic technique that involves the observation of motion of small particles in an electric field with a microscope, such as RBCs, neutrophils, bacteria, etc. The suspension is contained in a closed system composed of a thin-walled section for optical observations and of suitable electrode compartments. On the other hand, moving boundary electrophoresis is carried out in a solution without a supporting media. The sample is dissolved, the buffer and molecules move to their respective countercharge electrodes. The second type, that is zone electrophoresis, is a method in which an inert polymeric supporting media is used between the electrodes to separate and analyze the sample. The supporting media used in zone electrophoresis are absorbent paper, gel of starch, agar and polyacrylamide. The presence of supporting media minimizes mixing of the sample and makes the analysis and purification of the molecule from the gel much easier than the moving boundary electrophoresis. The gel electrophoresis is the best example of zone electrophoresis. Now let me enlighten you with the general techniques of zone electrophoresis. It includes paper electrophoresis, cellulose acetate electrophoresis, and gel electrophoresis. Firstly, we will discuss paper electrophoresis. It is a commonly used electrophoretic method for analysis and resolution of small molecules. This method is not used to resolve macromolecules, for example proteins, because the adsorption and surface tension associated with paper electrophoresis usually alter or denature the macromolecules, causing poor resolution. Two different methods are used routinely to apply samples to the electrophoretic paper. In the dry application procedure, a sample of solutes dissolved in distilled water or a volatile buffer is applied as a small spot or a thin strip of line drawn with a pencil called origin line on the paper. Appropriate standards of known compounds are applied at other locations on the origin line. If the electrophoretic migration towards both poles of the system is anticipated, the origin line should be in the center of the paper. If migration in only one direction is anticipated, the origin line should be near one end of the paper. After the solvent containing the samples has evaporated, the paper is dampened with the electrophoresis buffer, either by uniform spraying or by dipping and blotting the ends of the paper so that wetting of paper from both ends meets at the origin line simultaneously. In the wet application procedure, samples dissolved as concentrated solutions in distilled water are applied to paper dampened with electrophoresis buffer. After the sample is applied to the moistened piece of paper, the paper is placed in the electrophoresis chamber so that both ends are in contact with reservoirs of the electrophoresis buffer at the electrodes. If the origin line is not in the center of the paper must be positioned to allow maximum migration towards the correct electrode. After the chamber is covered or closed to protect against electric shock, an electric field is applied to the system. Application of the electric field and the resultant resistance to current flow in the buffered paper generates heat. This is the greatest source of difficulty with paper electrophoresis. Heat dries the paper, which in turn leads to more resistance to current flow, which causes greater resistance and so forth. Even if paper drying is prevented, heating will change the current flow and resistance properties of the system, which will distort the migration of molecules. Because of these difficulties, modern paper electrophoretic systems have been designed to compensate for potential heating problems. Secondly, we will discuss cellulose acetate electrophoresis. Cellulose acetate as a medium for electrophoresis was introduced by Kohn in 1958. It was developed from bacteriological cellulose acetate membrane filters and is commercially available as high purity cellulose acetate strips, which are thin and have a uniform micropore structure. Although paper electrophoresis is still the choice for routine fast diagnostic analysis, the resolution of a given protein might suffer because of substantial adsorption on paper. This disadvantage of paper is completely taken care of if cellulose acetate strips are used instead of paper. Additional advantages of cellulose acetate are: it is chemically pure, it does not contain lignins, hemicelluloses or nitrogen. Cellulose strips are translucent and this makes them suitable for direct photoelectric scanning for separated bands of components. Because of the very low content of glucose, these strips are suitable for electrophoresis of polysaccharides. Upon staining by Schiff's reagent, background staining is negligible. Cellulose acetate is not hydrophilic and thus holds very little buffer. This property is conducive for a better resolution in a shorter time. Lastly, we will discuss gel electrophoresis. In all the types of electrophoresis discussed so far, free electrophoresis, paper electrophoresis and cellulose acetate electrophoresis, charge on the molecule was the major determinant for its electrophoretic mobility and ultimate separation from the rest of the molecules. The gels, however, are porous and the size of pores relative to that of the molecule determines whether the molecule will enter the pore and be retarded or will bypass it. In gel electrophoresis, molecules are separated in aqueous buffers supported within a polymeric gel matrix. Gel electrophoretic systems have several distinct advantages. First, they can accommodate larger samples than most paper electrophoretic systems and so can be used for preparative scale electrophoresis of macromolecules. Second, the character of the gel matrix can be altered at will to fit a particular application. This is possible because the gel enhances the friction that governs the electrophoretic mobility. Low concentrations of matrix material or a low degree of cross-linking of the monomers in polymerized gel systems allow them to be used largely as a stabilizing or anti-convection device with relatively low frictional resistance to the migration of macromolecules. Alternatively, higher concentrations of matrix material or a higher degree of cross-linking of monomers are used to generate greater friction, which results in molecular sieving. One of the most commonly used among these procedures is sodium disulfate polyacrylamide gel electrophoresis (SDS-PAGE). It is the most common means of analyzing proteins by electrophoresis. SDS is a detergent which denatures proteins by binding to the hydrophobic regions and essentially coating the linear protein sequence with a set of SDS molecules. The SDS is negatively charged and thus becomes the dominant charge of the complex. The number of SDS molecules that bind is simply proportional to the size of the protein. Therefore, the charge to mass ratio should not change with size. In solution like water, in principle, all different sized proteins covered with SDS would run at about the same mobility. However, the proteins are not run through water. Instead, they are run through an inert polymer, polyacrylamide. The density and pore size of this polymer can be varied by variation in concentration of monomer and of cross-linking agent. Thus, the size of molecules that can pass through the matrix can be varied. This determines in what molecular weight range the gel will have the highest resolving power. In addition to this, I would like to explain some specialized electrophoretic techniques which include isoelectric focusing, agarose gel electrophoresis, capillary electrophoresis, and pulsed field gel electrophoresis. Let us first talk about isoelectric focusing. It's an electrophoretic technique that separates macromolecules on the basis of their isoelectric points. Isoelectric points are the pH values at which they carry no net charge. As with SDS-PAGE, this process can be carried out in a slab format. A pH gradient is established in the polyacrylamide gel with the aid of ampholites, which are small polymers, approximately 5000 Daltons, containing random distributions of weakly acidic and weakly basic functional groups. For example, carboxyls, imidazoles, amines, etc. A polyacrylamide gel containing these ampholytes is connected to an electrophoretic apparatus that contains dilute acidic solution, that is H positive, in the anode chamber and a dilute basic, that is OH negative, solution in the cathode chamber. When voltage is applied to the system, the current flow will be largely due to migration of the charged ampholyte species present in the gel. The ampholytes migrate towards either pole in a manner consistent with their charge distributions. Ampholytes with lower isoelectric points, for example, those that contain more carboxyl groups and have a net negative charge, migrate towards the anode, while ampholytes with a higher isoelectric points, for example, those that contain more amine groups and have a net positive charge, migrate towards the cathode. Eventually, each ampholyte in this system will reach a position in the gel that has a pH equal to its isoelectric point. When this occurs, these ampholytes carry no net charge and will no longer migrate in the gel. The net effect is that after a sufficient period of electrophoresis, the population of ampholytes will act as local buffers to establish a stable pH gradient in the polyacrylamide gel. This pH gradient forms the basis for the separation of macromolecules that are present in the system or are subsequently introduced into the system. Another one is agarose gel electrophoresis. It is the principal technique used to determine the size of high molecular weight nucleic acids, DNA and RNA. Agarose is a long polymer of galactose and 3,6-anhydrogalactose linked via alpha-1,4 glycosidic bonds. This material is readily isolated from seaweed. Agarose polymers may contain up to 100 monomeric units with an average molecular weight of around 10,000 Daltons. Agarose gels are made by dissolving the white agarose powder in an aqueous buffer containing EDTA and either tris acetate, that is TAE, or tris borate, that is TBE, as the buffering species. When the sample is heated to just below boiling point, the agarose powder dissolves in the buffer to form a clear solution. As the solution slowly cools to room temperature, hydrogen bonding within and between the polygalactose units in the solution causes the formation of a rigid gel with a relatively uniform pore size. The pore size of the gel in polyacrylamide gels can be controlled by the percentage of the agarose dissolved in the solution. A high percent agarose gel, say 3% weight upon weight, will have a smaller pore size than a lower 0.8% weight upon weight agarose gel. The percent of agarose to be cast in the gel will be determined by the size of the various molecules to be resolved during electrophoresis. The smaller the molecular weight of the molecules to be resolved, the higher percent agarose, that is smaller pore size, the gel should contain. The third one is capillary electrophoresis. Capillary electrophoresis can be used to separate ionic species by their charge and frictional forces and hydrodynamic radius. In traditional electrophoresis, electrically charged analytes move in a conductive liquid medium under the influence of an electric field. Introduced in the 1960s, the technique of capillary electrophoresis was designed to separate species based on their size to the charge ratio in the interior of small capillary filled with an electrolyte. Capillary electrophoresis offers the advantages of extremely high resolution, speed and high sensitivity for the analysis of extremely small samples, but is obviously not useful as a preparative method. It is especially useful in the separation of DNA molecules that differ in size by as little as only a single nucleotide. Because of its high resolution, capillary electrophoresis is the basis of separation of polynucleotides in some of the newer designs of DNA sequences. Capillary electrophoresis can also be adapted to the separation of the uncharged molecules by including charged micelles of a detergent such as SDS in the aqueous electrophoretic medium. Last one is pulsed field gel electrophoresis. It is a technique used for the separation of large deoxyribonucleic acid, that is DNA molecules, by applying to a gel matrix an electric field that periodically changes direction. While in general, small fragments can find their way through the gel matrix more easily than large DNA fragments, a threshold length exists above 30 to 50 kilobase pair, where all large fragments will run at the same rate and appear in a gel as a single large diffuse band. However, with periodic changing of field direction, the various lengths of DNA react to the change at differing rates. That is, larger pieces of DNA will be slower to realign their charge when field direction is changed, while smaller pieces will be quicker. Over the course of time, with the consistent changing of directions, each band will begin to separate more and more even at very large lengths. Thus, separation of very large DNA pieces using pulsed field gel electrophoresis is made possible. That was all about electrophoresis. I hope you have understood it well. Thank you and goodbye.